This is where we start to get into the levels of our actual immune system. Complement. This is an innate system of serum proteins that recognize and neutralize specific patterns within pathogens. Again, because this is innate, these are patterns that we're all born being able to recognize. Phagocytes are cells that then eat the extracellular material. These can be particles, viruses, general proteins floating through your body. It can even be dead or dying cells, or cells that are particularly tagged with antibodies. Antibodies themselves we'll cover in a little more detail in the next slide and in lots of detail in later lectures. They are part of the adaptive immune system. In general, they tend to target specific elements for phagocytosis. But one of the things that they can also do is neutralize a pathogen by binding to its effector region. And we'll get into that strategy again later on. Now, assuming that the pathogen survives this level of defense, it then has the potential to go inside the cell and become intracellular. There is then another series of defenses that we have, including what are called pattern recognition receptors. These pattern recognition receptors signal to warn other cells that there's a pathogen present. They also initiate internal cellular defenses, and if they see fit, they can also initiate the death of the cell. We also have what are called effector T cells that can then come, when a cell is warning that it's infected, the effector T cell can come and kill it, and hopefully protect other cells from becoming infected. Now, before we go much further, I need to cover some specific terminology. This is an antibody. This is the structure of an antibody. You can see in blue the constant or effector region. This determines what the antibody does. Now, the red part is the variable region. This is what makes an antibody part of the adaptive response. You'll note that there are actually two arms to this antibody. Many times, antigens are thought to bind within the center of this V region, when actually, because there are two arms, they can bind two different antigens. Now, I keep saying the word antigen. What is it? Well, an antigen is anything that can generate an antibody. This is most commonly thought of as protein, but it can also be sugars, fats, in fact, a great many things. Now, there's a specific type of antigen called an immunogen, and this is an antigen that can induce antibody production. Again, not all antigens are immunogens, and this is something that we'll cover when we get to vaccines. How do we generate the appropriate antigens when we don't have an immunogen present? This is a diagram more of what we think of as protective immunity. It's a map of the adaptive immune response. You'll note that this is also looking at antibodies. The other arm of the adaptive immune system, T cells, act the same way. Introduction of antigen A at day 0 leads to a response 4 to 7 days later. This is the initial response. And then upon subsequent challenge with antigen A, we see a greater and faster response. This is the protective nature of the adaptive immune system. This is why we don't get sick a second time around with the same virus or pathogen. So I've thrown a lot at you. Let me give you some points to think about. These questions will be posted to the discussion board. And so I want you to post a thoughtful response. Doesn't have to be correct, just whatever you think is correct. I just noted that the adaptive immune response takes 4 to 7 days to actually respond. Why do we have this delay? And is there any protection during this time? How does the immune system keep a pathogen in check during this time? And most of the time that you're sick, your immune system is actually causing the symptoms. What is the advantage to this? Why does this occur? And how? How does your immune system make you sick? What do you think is your immune system doing to make you sick? Now, immunity is generated against multiple pathogen components. How can the immune system do this? And why would we want it to? And why wouldn't we want it to? And you'll have to take on my word that it's not always the same components between individuals. So, as an added bonus, how is this variation generated? And why would we want variation? And why wouldn't we? Again, these questions will be on the discussion board. Please provide a thoughtful response by the next lecture. So to summarize, our goal as immunologists is to maximize protection while minimizing the pathology. There's a variety of potential immune targets, which you should keep in mind include our own cells. So think about why we would or would not generate an immune response to our cells. An immune response is a last line of defense and made up of innate and adaptive components. These two components work together, and by the end of the course, I want you to be able to tell me how. And so next, we'll be talking about immunity as a system, what the cells of the immune system are, as well as the major anatomical regions involved in the immune system.